Hi, thank you for checking out sciencevideos.org, where all the latest science videos. Today we're talking to Professor Per Ahlberg, talking about his paper on the evolution of enamel. So Per, what can you tell us about your paper and your work?
All right. Okay, so what we've been looking at is the origin of enamel. I mean, enamel, of course, is a familiar tissue from our own bodies, what covers the surface of our teeth, and it's the hardest tissue produced by the human body. In its sort of final form, it's very largely just mineral, but it's deposited on a matrix of 3 unique enamel matrix proteins that don't appear anywhere else in the body. Now, we were interested in finding out where this tissue had originated, because just because you have teeth doesn't mean to say you have enamel. Sharks, for example, have teeth, but they're not covered with enamel. They use a different surface tissue. What we do know, however, is that early fossil fish and some living fishes don't just have teeth in the mouth, they're also covered on the body surface with little skin denticles, which might sit on big bony scales or just sit loose in the skin, which are made of dentine, which is the core material in our own teeth, ivory basically, but which can also be covered with enamel or a similar sort of substance. So we wanted to know where enamel had originated and in which groups really get it. There were kind of 2 questions here. One was whether enamel actually starts out on the teeth or somewhere else in the body, and the other was whether a peculiar tissue you get in some living primitive ray-finned fish, like the garpike, Lepisosteus, or Polypterus, which is another of these primitive fish, a tissue called ganoine, whether this stuff is really enamel or not. There's been a lot of debate about that. The stuff looks a bit like enamel, but there are detailed differences in the microstructure. So we pulled together 2 sorts of data here: fossil data from early vertebrates and genomic data from the spotted gar, Lepisosteus oculatus, which has recently been sequenced. And the first thing we did was we looked at the gar genome to see whether it has these enamel matrix protein genes. And it turns out that, yep, they're present in this fish and they are expressed in the skin. Well, 2 of the 3 genes are present in the fish, and they are expressed in the skin, which is where ganoine forms. So this really supports the idea that ganoine is a true form of enamel. Because bear in mind, we don't use these proteins anywhere else in the body, okay? They're unique for enamel formation. The other thing then we did was we looked at some early fossil fish, in particular a form called Psarolepis from China. It's about 415 million years old, and it's one of the earliest bony fishes. Turns out this guy is covered with enamel on the scales. It's got little denticles on the scales, they're covered with enamel. It's got denticles on the skull bones, they're covered with enamel. But the teeth carry no enamel, they're just naked dentine. It's beautifully preserved material. You'll have these little enamel-covered facial denticles physically next door to teeth with no enamel. So it's not like some sort of preservational damage thing. The animal is really like this. And in fact, we have another early fossil fish from Sweden, a thing called Andreolepis. It's a little bit older still. The material is more fragmentary, so you have to be a bit more careful. But again, it looks like this one has got enamel on the scales, but no enamel on the skull bones this time, and no enamel on the teeth. So we've got 2 stories coming together here. One is that enamel on the body surface seems to be pretty widespread. It's there in lobe-finned fish. These are the fishes that kind of give rise to land vertebrates. It's there in ray-finned fish because ganoine, which they have, is a form of enamel. But these 2 groups are sister groups, okay? So you've got lobe fins leading towards us, ray fins leading to modern bony fish like zebrafish or what have you. All of these are covered primitively with enamel on the body surface, but their teeth, it seems, do not carry enamel. So the story seems to be then that enamel originated as a protective tissue on the skin surface, and only rather later did it colonize the surface of the teeth.
Okay, so thank you for that lovely overview of your work. I couldn't have done it by myself. A few questions. So why do we think that the enamel moved from the skin and the scales to the teeth?
Well, that's a really interesting question. We don't really have a good answer. I mean, obviously, once you've got enamel on the teeth, it's very useful because it's a much harder surface of the teeth than just dentine. It makes the teeth sharper, harder wearing, all this sort of thing. Why it moved is difficult to say. We want to look into this because a reasonable guess would be that, obviously, we've got a set of genes that tell you how to make enamel, and these are employed wherever enamel pops up. Excuse me. The key question is, where is this activity initiated, and that's going to be a matter of the enhancers that control the activation of the enamel matrix protein genes, probably, and maybe some other genes besides. So we want to look into the organization of the sort of the enhancer regions for these genes. A reasonable guess might be that new enhancers popped up that simply conferred enamel activity onto a new part of the dental skeleton, perhaps in a sense with no particular reason. You know, it's just a sort of chance thing that happens. But once you've got this tissue, it's of course very useful, and you don't want to get rid of it. So that's probably the best guess we have.
Yeah, so as well, do you think that enamel has come from a single origin point? Do you think it was one species that evolved the enamel, and then it was like an arms race, the evolutionary arms race, and more species started to develop enamel? Or do you think it's a dynamic thing? Do you think it's evolved more than once throughout history, or do you think it's just this one event and it's stuck?
Now, I reckon it's in its origin is basically one thing. I mean, of course, you know, it wouldn't have popped up, you know, boom, just like that. It will have been a process, but something relatively rapid. Because, as I said, bear in mind, what's distinctive about enamel is that it's built on the basis of these unique proteins that aren't used anywhere else. Now, for comparison here, look, when you form a tooth in any sort of animal, you make a fold in an epithelium, okay, and inside that epithelium, you've got a mesenchyme of cells, and the mesenchyme is going to deposit dentine, and it always grows inwards. And the epithelial cells, well, what they do kind of varies. In some fish, you find no surface layer on the dentine at all, or like on the teeth of Psarolepis, and it looks like the epithelium just kind of sat there. But if you look at, for example, sharks, they make, as I mentioned, this alternative tissue, enameloid, on the surface of their teeth. And what that's about is that the epithelium produces a mineral component, but not the matrix. It's just mineralizing the surface of the dentine matrix. So that's compositionally quite different. It's got collagen and things in it. So at a reasonable guess, the ability of the epithelium to somehow hypermineralize the surface of the tooth is an ancient thing, and that's been redeployed in several locations. So that might be our really deep basis for how you can make enamel. But the specific thing about enamel, the combination with these proteins that aren't used elsewhere, and the consequent complete separation of enamel from the dentine matrix, so you have this totally sharp boundary between the two, that's a unique thing, and realistically it has only arisen once. But I tell you what, though, there's a very curious further twist to this story, which we don't yet fully understand, because there are some, in the human body, or in the mammal body, there are some additional proteins that are actually very closely related to enamel proteins, and they're the proteins that go into milk. This is just insanely bizarre, but it looks as though much later the enamel proteins may have been redeployed in a secondary role, not to do with biomineralization at all, but with the initiation of milk production. This is really interesting. We want to look into it, but it's not something that we've really kind of dug deep into yet. I just want to flag it as one of those kind of weird things that's out there.
Yeah, so that's kind of what I'm leading on to next. So what is the next step of this research? Where do you plan to go from here?
Right. Well, I mean, we obviously want to understand better the actual molecular basis for enamel production, and in particular how that is regulated, and we're pursuing that. But additionally, this has proved to be a really fruitful way of pursuing the evolution of hard tissues. I think this is one of the first studies where we haven't just tried to integrate data from paleo and, say, genomics, which of course has been done before, but really using new data from both sides of the field, rather than, you know, having new data from one area and then looking at it in the context of already existing knowledge from another field. So we want to continue with that, and there are other hard tissues to look at here. You know, bone itself, dentine, that's going to be interesting. And there are peculiar tissues, like, for example, acridin, which we get in the tips of the teeth of rayfin fishes. It's a kind of hypermineralized dentine that just sharpens the tooth tip. But the point with all of these is that mineralized tissues, by their nature, tend to have a really good fossil record. The genetic basis for these tissues is moderately complex, but not stupidly complex so as to be difficult to investigate. And so we have a really fruitful arena here for future work. We will continue doing the same sort of stuff with other hard tissues and see where we fetch up. Okay, so thank you, Per. You've been brilliant. You can find a link to the paper below this video. This paper was published in Nature Letters, so it's a really good read if anyone's interested. And thank you very much for your time, Per. It's been lovely to have you. All right, very good. Thanks very much indeed. Thanks for the invitation.